Okay, now we'll come to my topic. Audible, madam?
Yes, sir. Yes, sir.
Okay, my topic is biofuel for sustainable future. This is actually a billion-dollar business. People think India has an ample scope for production of biofuel. Even UK has started some unit in Karnataka. So that was the scope for biofuel. Probably you know I will go and explain you why it is a sustainable future, and it also reduce the pollution or greenhouse gas emission will be reduced, and there's a lot of benefit using the biofuels. So there are different way also. You will save the oil, saving in action. Better fuels is biofuel. You can save 50% of the fuel, and the biofuel is the future fuel, because no other way we can run our vehicle with what you are running today, petrol, diesel. Even coal will be exhausted. So is other alternative. When you come to the world energy requirement, okay, look at the demand for the world energy demand. See, every year is increasing. So 2050 we require 5 to 30 TW. That's a tegawatts requirement, actually the requirement. So that's at present we are getting energy. Total energy requirement demand is so much, but it increases to the world. This is actually the world energy demand. Out of that, we developing countries also require. This is actually increasing order. Look at that. I think developing countries may need. Already you are reached there in 9 TW tegawatts. So this is a gap. When you whatever the energy existing today, we have around 14 TW is the gap 2050, 33 will be. What will be the alternative? This I think the world fuel mix like this. The majority, more around 40% from the oil, and 20% in the gas, 22% from the coal, and also nuclear. Actually, that was a year earlier, it is higher. Renewable was less. Renewable, it is reaching around 9, and it is around 7% of the total world energy requirement. When you compare that, 85% is the fossil fuel. What is the fate of fossil fuel? We get it in the future. So this is the again. What is the demand and the projection for 2000? Anywhere is increasing, but even renewable energy is also increasing. Look at this. So there's a trend of increasing renewable energy. Natural gas is increasing. Petroleum, I think everything we need is increasing rate. This is the total quantum in the terms of BTU is the unit of energy requirement, world energy consumption. Now that means it is very clear that the energy demand is very huge and need energy. But when it comes to the fossil fuels, the fossil fuel is the graph explaining about how the fossil fuel is depletion. What is the rate of depletions? So earlier it is actually the rate of depletion is very high. So look at that. This rate of growth rate is 2.2. So we'll have around 2050, we will not get any coal at all. So this curve represent the coal, and it is increasing. So that is the deadline, 2068. And when it comes to the other fuels, like other fuels, it's also declining. So it is also declining, like a natural gas. So natural gas is also declining. So from the look at that, there's a 2046 is the world. Actually, what I'm giving this date is year is the world date depletion, but for India it is still 20 years less. So maybe we get around 30 to 35. 30 to 35. So that's when you come to the oil. See, look at that. Oil is also around 2030. But maybe will not get a fuel for India after 2030 or 35. So this is the way we get, because you know what is fossil fuel? All of you know that fossil fuel nothing but a deposit. Now you are taking the deposit from the earth crust. The day when you know when you have money with your part a packet, it just keep on removing, will not get. It cannot be renewable also. The fossil fuel what today you are using coal or gas, natural gas or petrol, diesel, or come under the oil or kerosene, all come under the oil structures. So that is non-renewable. We cannot get back. Once it is removed, it is moved. So that's why it is a non-renewable fossil fuel, and we do not have. Just imagine when you do not have a petrol and diesel, how to send our children. I think most of them are young and energetic. The student, you can look at that. This is the 2040 or the 50 vehicles. You can say 2040, you have to send your children like this. So you have to use arts cart or bullock cart, whatever. So this is the way. But you cannot depend on, because the when you use this the kind of old traditional way of vehicle for transportation, it is a lot of time consuming. You already adjusted to the speed and the time and other thing. We cannot do the things like that. So this is the reason why the biofuel is required. No doubt, I think all of you convinced that biofuel is required. Okay. So now, what are the different types of biofuels? When you come to the biofuel, we can classify it into three groups: solid, liquid, and gaseous. So those are derived from the biomass. It's very important. Those are derived from the biomass, either from the living organism or from the metabolic byproducts such as a methane from manures from the cows. So these are the biofuel made up out of the organic materials, which made up, in contrast to the fossil fuels such as coal, petroleum, and others, are come from long dead plant and microorganisms. The fossil fuels are formed because of the high biomass which are there earlier, and this again it is submerged with high. There's a lot of earthquake and other thing. The entire plant or microorganisms which are down to earth and with high pressure and heat, it converted into fuels. Come to the biofuels, solid and other thing. So these biofuels, which actually renewable, so those will be derived from the biomass. So those will be renewed from the time to time. We'll renew it on the quantum. So then the biofuel which are renewable when you come to the solid. So you take an example of any weed. So in a forest, usually during the summer there's a fire catching in the forest and burn lot of berries because of the weeds which grow in between the rows of the plants or in between the plants or the trees which are grown. So this is a heavy loss to the nature and adding. Then those biomass, and there is a one source of biomass for us. The second source of biomass now is agriculture waste. So for example, sugarcane trash or bagasse or maybe any coconut trees. Can you? Coconut trees. So these, like that, any biomass which are on weeds on roadside and antenna weeds. So okay. So those, yeah, someone is talking, Jyoti. Okay, that's what on these materials, the biomass means which are actually weeds and everything directly. You make them directly used to boil the water. Then you get a steam. After the steam, and it will goes to the generation of electricity, that is called co-generation. So in most of the sugar factory, they will generate their own electricity and run the factory in sugar factories. So that was an advantage. So CMA we can do also, we can generate electricity. When I was the member of the Biomass Diversity Board as a coordinator, evaluated in the biomass availability in agriculture area like in Mandya district and Karnataka. So where I found that 75% of the electricity which are required for particular taluk or the district can be generate its own electricity using the biomass. So now we can take lantana weeds, whatever the weeds available, you cut into that or make some energy plantation also along the roadside, along the railway line or in wasteland and agricultural, whatever all around the bounds of the agricultural land, we can harvest it once in 6 years or 5 years and use them as a source of fuel and generate electricity. Or otherwise, another way around, you have a lot of biomass is a heavy and difficult to transfer. So such things we can, what you do, we can make a briquettes, a small briquettes. So in future, when you don't have a gas or something, you have to use the briquettes only. So there is another scope when you come to cooking your food. I will come back to that. So these briquettes can be used and generate electricity. So then we can do boil it, or you can boil it and cook the food. So that's a way the solid biomass can be used. The second one is liquid. They convert any biomass into various forms, either as a methanol or ethanol or as a butanol. So then we can use those things. And apart from that also, then oil seeds or waste oil can convert it as a biodiesel. So and either bioethanol or biopetrol or bioethanol or biodiesel. So this is called liquid forms of and also we called as even oil also, crude oil out of that under the anaerobic conditions, liquid oil or jet fuel. So another one is called jet fuel. It is actually the conversion of entire algal group into a jet fuel. Polycarbonate materials we can make it out. So that is the second form. The third one is gaseous form. Again, make a anaerobic fermentation. They convert them into manures or any industry, the urban liquid waste. So can be converted, or maybe distillery effluent from the distillery can be converted into methane or gases. Those gas can be used as a fuel. These are the way 3 forms we can get. And totally when you use this biofuel, there is no addition of carbon dioxide to the nature. So then no carbon dioxide addition and no question of rising the temperature and the question of melting of water bodies, and also finally the melting of the ice that resulting in increasing the level of sea. So if you not maintain properly another 50 years, so we cannot see the cities which are nearby the oceans or which are in the border area of actually the area. That is why I think beach area, whatever you get, that will be submerged. The cities which are there in the near to sea oceans will be submerged in future. So that's why the water, the carbon dioxide emission is also playing an important. The major biofuel producers by region wise is Europe, South America, and is the top most is that. When I was the PG student, the America is not in a list of biofuel production. Today it is number 1 in the world actually. And that time actually Brazil and India was the one which are producing bioethanol. That time America is not in the picture. What they did in America, now they will start converting corn and soybean oil into biodiesel, corn into ethanol. So what they do, the farmers themselves, they have a very big land holding. They will produce in terms of tons of million tons of or lakh tons of corn seeds. Those corn will taken to their own factory, where separate the corn into protein and also another is a carbohydrate. The carbohydrate will be converted to ethanol. The protein goes as a animal feeds, and that goes as animal feed protein part. So like that, they are bifurcating and selling this ethanol, bioethanol, to the industry, petroleum industry, where they'll blend with the petrol and this is called as gasoline. So like that, we'll see in next South America, Brazil will come in that picture. So that is a second place. So that also producing in South America, they can grow sugarcane. The next is the sugarcane can grow very widely in India. India has a lot of scope to produce bioethanol using the sugarcane. Again, the question arises when America start using ethanol, using the corn and soybean. Entire world was opposed it and commented on that, but the America never bothered about it. But what they did, They use all genetically modified herbicide-resistant maize. Here, the corn which can be produced out of that will be used for the ethanol. The production is so high. Technology now today, the 10% is blending must in case America. Where ethanol advantage is also there. When you use the ethanol blend mixed with the petrol, it acts as oxygenate and the efficiency of the fuel will be increased. And that's how the people now get benefit out of it. But India, you were earlier in the top 2, now it is in the, maybe you're going to below 70. That is a rank. The Europe is also producing and Australia says very less. And now only people in some country like palm oil is excess, where they're using palm oil. So they convert into diesel. And in case of Africa, they're using converting animal dung into methane gas and jatropha. Yeah, they're going very widely without any investment. So they're using jatropha seed and converting into diesels. So now you got it. The corn, sugar cane used for production of bioethanol, soybean, jatropha, palm oil used for the production of biodiesels. How to produce bioethanol means take the sugars, go to fermenters, take into the ferment, make a liquid, adjust the sugar concentration, add yeast cultures, and allow for fermentation for 15 days, 30 to 40 days. Then when after fermentation, distillate, the ethanol which are produced in that liquid will be evaporated, collected, condense it, and take the ethanol. That is called bioethanol. Again redistillate, get 100% ethanol. The next is jatropha oil, any oil, you take any even waste cooking oil, you take the oil out of by the soybean or the jatropha or palm oil. So trans-esterification, though that will be done under the process of sodium hydroxide and methanol as a catalyst agent and put it under the 40-degree temperature. The entire oil will be trans-esterified and finally we can collect the diesel, biodiesel, that is only fatty acid, and glycerol will be separated. Glycerol can be used for the soap and the total bioproduction around 81 billion liters now. So in that, 40 billion liters is from the America, North America, South America in 25, and palm oil in India, maybe not India. India cannot even affordable to use the palm oil. Very small countries like Indonesia and other, where the excess palm oil production they are using. And here the corn and soybean being used in care of Europe. And also in Africa, they're using jatropha and others. So the types of biofuel based on use, we can also classify it into bioethanol. I told you already, bioethanol is pure alcohol. It's the most ubiquitous of alternative biofuel making their way into people gas tanks. It can be made from various sources, even not only from sugars. We can also made out of cellulosic ethanol from sugarcane bagasse, waste product from the sugar processing, even wood, dust, sawdust. You can use the sawdust also. We can use the grasses. Anything we can be converted into. And also we can use the directly from sugarcane. Juice can be converted to ethanol, or the corn, sugars, or any product which rich in sugar can be converted to ethanol product. The advantage renewable ethanol is generated given sufficient crop yield. Domestic production reduce the dependence in foreign fuel. I think we India more than 70% we are depending on the foreign exchange. So if you do only our own culture, I think government of India now give a lot of initiation and give a lot of subsidy also to promote the ethanol production. Probably today's newspaper, if anybody read it, I think Gadkari, the Minister for Transportation, released the vehicle which can run with electricity and ethanol. The future vehicle is only run with the biofuel and the electricity. That is a blending, a mixed vehicle. Can either use electricity or you can use this one. So there's a new vehicle, it is released today. We can see the newspaper, we'll get a lot of information about that. It shows that there's importance. I'm telling why I'm telling it is an important future, only you can have a vehicle like that. So then ethanol cleaner burning than gasoline because it is oxygenate and 15% less greenhouse gas emission compared to just if you are burning. Advanced fuels, ethanol, make ethanol fuel from the waste like your food and whatever byproduct is also. These are the advantages. But there are some advantages. I will come back to that. Negative also. The ethanol derived from corn, wheat, sugar cane compete directly with the food supply and derive from the cost of other food. And this was the major against for user. But my opinion, I know this agriculture since my childhood. There is a lot of scope. This will not get any food problem at all. Because you look at that, the people who are eating your maybe grandfather, mothers, they're eating very more and doing a lot of energy work, physical work. Today, you look at your young generation, you are not spending much more energy. You need only for small, and even sugar is not at all required for us. Sugar can need not go for produce the sugar at all. Why? We need only the fruit sugar or sugar in vegetable or sugar in cereals or the pulses is enough for our body. No need to produce the sugar at all. The white sugar is dangerous actually, and it creating a lot of diabetic. India became world capital for diabetic patients. So the young generation is suffering because of they are not spending money, spending energy of body, or you are working, you're eating more than what you require. So no question of sugar cane. The beet sugar not at all comes in the picture. So you are producing excess corn more than what is there. If you go to transgenic, the yield will be doubled or more than more what is required, and the excess corn can also be used for that. This can be solved because number one, ethanol has less stored energy in gasoline, reducing fuel mileage between 50. No doubt in that. So, but that can be enhanced. Ethanol can be converted into butanol. The butanol can be stored easily, and you can immediately mix with the petrol and diesel and start. It currently requires more energy to create large quantity ethanol than it produces. However, still ethanol may be a magic bullet, but what they're telling more energy requirement for the crop. But instead of wasting the crop, so you can do things out. So converting standard gasoline engines run high blinds with costly. No, it is actually only small modification required. Recently, today, I told you also the blended type of vehicle, electricity and ethanol is released, and that will not cost much. It's also. Then I answered all the question to you. Next is the biodiesel. It can also produced in the oils, but now you are given example. Here is sunflower, seed oil, crude oil. This is how the extract of the oil will get a crude vegetable oil. Again, refine it, filter it, we remove all the dust which is there in that. Then go the transesterification using alcohol. And finally, we get the transesterification. Then we get a glycerol can be used for food industry, cosmetic industry. Then whatever the other part is called biodiesel can run onto the vehicles. And again, the carbon dioxide which is released out of it. Again, the crop synthesizes carbon dioxide taken back, and that's why no addition to the atmosphere. So exacting, this is how we can get the. Again, pro is there. Always is a butanol. Biobutanol is produced from the algae bacteria. Can be used directly in gasoline with no modification. Can be used existing pipeline. Can also be used. No, the octane level is very. There's a little, if any, loss in the fuel mileage. No corrosive to engine and pipelines. The domestic production reduces dependence on foraging. So these are that one. But disadvantage I looked at. Feedstock are required for production, although non-food stocks and genetic bacteria can strain in all this. So what is there? I think when you start eating all genetically modified food, nothing will happen when you modify the bacteria, produce more of ethanol or made biodiesel. Although we can also use our food products also. And there is excess oil, okay, palm oil, or maybe oil content in the soybean. So then we can reuse. Production cost is high because we know that. But today the cost of 1 liter of diesel in our India, in our own university campus, we are selling, it is around 60 rupees. Look at the diesel price is increasing. Earlier what was there, the production cost, today it is decreased also. It is coming down, and every day it is coming down. The biogas is another energy, gas produced by anaerobic digestion. So that has to be done. Earlier the government of India has promoted for biogas production in rural area. The day will come when no gas for cooking your food. The biogas, particularly methane gas unit, is very good. So when I was the 7th standard, I used to have a biogas plant in our home. They are used very well, and it's a good for us also. And we get a very good manure also after the biogas, whatever the cow dung, after manure, we get a very good. And I think the waste management, there's a lot of fund available in that. The youth can take some project and convert the waste into value addition. All waste, whatever the municipal sewage waste, food waste, and can be converted into biogas and mainly of methane and also. And this can be used for vehicle fuel or generate the electricity. Burn directly for cooking, heating, lighting. So in my home, in our own area, we used to this for cooking, and also there is a lighting also available. I used to enjoy that during my childhood. So that again it will come back. Only thing the time has taken. Maybe another 5-10 years, you have to go back to the back. So again, I told you this is a total cycle how the biogas can be used very well. And we have the animal, or maybe you can take waste from others, light, photosynthesis, vegetable biomass can be used, and the organic waste from the areas like the urban area. So everything come for anaerobic digestion. So then we will get a biogas, can even generate electricity, heat, and again they take carbon dioxide, what is released, they take oxygen. Then again, the whatever the leftover is a good fertilizer. Again, this fertilizer can be used to grow the crop. And again, you can use it. There's no addition of carbon dioxide to the atmosphere, no greenhouse gas effect, no increase in temperature. So this again, I told you already. We use the cooking and heating and lighting. This is a lighting actually, and we can also use for generation of electricity. And slurry is a good biofertilizer used for the crop growth. And these are the waste. You can use the kitchen waste, animal manure, and agriculture residues, and it is converted into. And is eco-friendly. Generation reduce soil and water pollution. And biogas generation produces organic fertilizer also. It is simple, low-cost technology, encourages circular economy, healthy cooking alternative for the. If any unemployed, we can start a small unit in our own area and start working and generate electricity or whatever. That will be really helpful for. And a few technological advancement and government has now been giving. When start using this technology, certainly will get the contains impurity can be using the filters. We can remove the effect of temperature on biogas. No doubt that the temperature should be around 37, and in some of the states in India have a 37-degree temperature. 35. Less suitable for dense metropolitan area because need lot of space. So this can be widely used in rural. So next is this I told you the new product called biobutanol. It is not there earlier. Now we can, while less well known for 4 biofuel future, biobutanol was the most promising. Is a simply isobutanol derived from bacteria or algae, much like measles. However, the beauty of biobutanol is that it can potentially be directly used as in a standard gasoline engine, no molecule. So this is a new fuel, so that can be used very well. And again, this is renewable. Can be recycled. Domestic production reduces dependency on foreign fuel. Reduce the tile pipelines with emissions and other. The people telling biodiesel is costly now, it is not costly now. Earlier it is a costly, expensive. And when the diesel was 40 rupees, now now near to biodiesel, diesel is near to 100. It is not at all costly now. Only thing people should do cold weather can gel when the temperature drops, causing fire injection problem. Not that we can have a modification in engine, or you can put some heating unit below the tank of the biodiesel, then can be done. Several diesel engines can run on biodiesel with a minimum of modification. This is the. So now I'll come to the again the biofuel based on the crop or resource, or we can say the input. They are using it. Based on that, they classified into different generation. It is a 5th generation actually is there. So I'll explain only up to 4th generation. The first generation, people start using the edible. Cassava, sugarcane, molasses, palm oil, and groundnut oil also. So all of you know that actually the first diesel engine is run in Europe when it invented. It is with a groundnut oil. The sixth, and also this is for biodiesel production, and again sweets were gum used for the production of bioethanol. And this is the edible oil. These are the edible one, and these are the non-edible tetrahydrocanol. So can be used widely. And second generation based on the cellulosic ethanol or waste biomass from the agriculture sector, like sugar cane, grass, leaf, whatever. And third generation is using the algae as a First generation people, I told you there's a lot of competition for food security versus fuel. My opinion, with the biotechnological invention, we can increase the production. I'll tell you one example of sugarcane. The average sugarcane yield in India, it is around 25 ton per hectare. But when the potentiality is there, 100 ton per hectare. So some farmers grow even 125, but on average, we can grow 100 ton per hectare. But actual average now it is 25. That means when you get a good price, the people certainly grow more, and you have a lot of scope to that. There's a first generation will not at all a problem. The maize I told you already, sugar beet, whatever will not be a problem for. So the first generation. So the another one is called syngas, is also new concept, and I'll come back to the second generation. Is not a problem now. It is a non-edible fuels which cannot be compete with the food security and eco-friendly. And this one actually second generation, no comments on that. People are using very widely. So this is ethanol from biomass. So how the people are thinking during my visiting professor, as a visiting professor to Sweden, I visited Umeå University. In that actually they already started biomass sawdust. You know, Sweden is known for forest. It has a forest area of more than 60%, 60 to 70%. They will cut the trees and making the furnitures and selling to America and other countries, Europe countries, where actually there are a lot of sawdust generation. People start using the sawdust and producing ethanol. They already have the unit. Even the unit is not viable, but still they're running because they knew that the cost, one or the other day, the outbreak of the cost will come back, and the technology will be useful in the world. Probably when that start working and improving a lot of things on that in your factory. But now what I feel it, they will have expertise in technology. And in future, they'll give the technology to the world, and they'll making a lot of patenting also. So this is how the people in other countries work, make the product patenting. So and then like country like India, we cannot affordable so much of research, amount towards the research. So that was the main constraint. Then other countries they are patenting and sell their technology with high cost. So that is happening in all the issues, like in case of genetically modified crops, or in case of any chemicals, all other things which are patented. Even in pharma also. It's not only agriculture, in all industrial sectors, in all equipments, whatever they are patenting. So they but whenever start, they will start with the even negative, they'll not bother, but the technology being transferred, and finally they'll get a lot of, the country will get money. But India, like other countries, they have to buy the technology which are the cost. The third generation is algal fuel, is also called ILA, or third generation fuel, is a biofuel from algae. The way of algae is low input, high yield feedstock to produce biofuel. It produces 30 times more energy per hectare than the land crops such as soy. So this is when you have a land area constraint, so we can go and use the second and third generation fuels are also known as advanced biofuels. So this is how the biodiesel production from the algae. So this is in a wasteland, or we can use any land, desert land area can be used. We cannot grow any crop. We can just put all the cells and then harvest. This is the mass of algae. Free extraction they will do. And then you can go for the, after getting oils, the lipids are available, then go for transesterification, then go to the bio. And apart from that, so whatever the fat will be converted, remaining will be as a value added product. We can also produce some proteins and other. So this is another picture explaining you how the algae in the sunlight, carbon dioxide, water can produce algae. It's a very fast way. Then extraction of algae to first produce 70 to 70% of algae from the plant. So then this oil solvent used to separate the sugar from the oil, and solvent then evaporate, and oil is ready and used to directly diesel engine or refined further into. So see how the area people are using, lot of area being used to cultivate the algae, both horizontal and also vertical. These are the vertical. The fourth generation, we make use of novel synthetic biology tools and are just emerging at the basic research levels. So the fourth generation biofuels are produced by designing photosynthetic microorganisms to produce photobiological solar fuels. By combining photovoltaics and the microbial fuel production, it is called electrobiofuels. By synthetic organisms specifically tailored for production of desired high value chemicals. So production currently based on fossil fuels and also biofuels. The fourth generation technology combines genetically optimized food stack, which are designed to capture large amount of carbon and genomically synthesized microbes. Here the microorganisms which are modified to synthesize a lot of fixed carbon dioxide, efficiently make a fuel. The capture sequestration carbon, because they render the fourth generation. The biofuel is also called as carbon negative source. The process technology in biohydrogen. You know, the biohydrogen is another scope. Hydrogen is also sometimes called as a fuel cells. This technology already is under patenting, and one of the Indian origin scientists working at US has developed the fuel cell, and the cell is the size of a refrigerator in a home. You can take that fuel cell box to your refrigerator cell or the refrigerator to your home. That box will be installed in your home, similar to the refrigerator. Then electricity, you can get electricity. So there is a new generation, and now it can produce hydrogen. Hydrogen is fuel cell is very important now. One is the biological way, another thermochemical way, and electrochemical. Biological way, the fermentation and also and the thermochemical, these are the things: gasification, partial oxidation, steam reforming, cracking, and fireless. The fireless is one of the important technology in biofuel energy. And the Indian Institute of Science at Bangalore, they are pioneer in the biolysis sector. So electrical chemical way, electrolysis, and also photoelectrolysis. So this is the fuel cell. It's a device that converts chemical energy from a fuel into electricity through a chemical reaction, which with oxygen or another oxidized. The hydrogen fuel cell stack, I told you example. So this is how the fuel cell stack has been developed by Indian scientists at America, and is a patented also. Maybe in the future we get this hydrogen fuel cells, and it can be generated electricity and can be used for the. And the overview of the total biofuel production. Start with the first generation, just I'm explaining, using cereals, starch, sugars, wheat, soybean, sunflower, even potato, maize, everything can be used. That is called to convert these products into the compost generation. Second generation, where it can manure, and also you can use the straw, paddy straw, and also municipal waste, industrial waste, and manure, and the crude glycine also to produce second generation. Third generation is algae, and fourth is the specifically designed microorganism, genetically engineered crop, and algae can be widely used in the fourth generation. So this is the young generation I'm telling. I have a lot of scope to this, and technology available at this stage, we can start produce commercially also. If you take example, second generation is the best way, because it's not much interference with the food security. So we can start as an entrepreneur. We can start this, and we can also work out on third generation. The fourth is for research purpose, ideations. Biotechnology, making the bioproductions, which is also helping for the biofuel production. I told you very important. Crop can be modified. So biotechnology approach for cell wall modification. I give example of one of the modified called alfalfa. It is also called as lucerne. It is a legume plant and very good for, actually this plant has been used widely. Lucerne is also called as ars gram. Actually ars has been used. This fodder, a legume fodder, widely used to feed the ars, because it gives a lot of energy for the running race or whatever the work they're doing. So what they did in that, actually it contained a lot of lignin content. So they now, with the genetically modified, the lignin content has been reduced. Even if you reduce the lignin content in the cellulose, you can produce a lot of, convert very easily. Lignin is one of the calciferant. It's restricting the process of fermentation. So when you reduce the lignin content, easily we can convert. Enhancing the biomass yield, another very important. I told you enhancement in the yield of maize, soybean, everything is done because of the transgenic, either herbicide resistant or pesticide resistance, whatever. So even that can be done with the biomass. And delaying the flowering in some plant, get a more biomass. That is another way of approaching. Also be, and also you can also have a more of carbon binding molecules bind to polysaccharide surface, and that also be another way. Reduce the lignin content here, and reduce the flowering, increase the biomass by the photosynthetic efficiency plant, increase improved growth, and that is also be done with the enhancing here. Similarly, I think these are the way how the people have done on some research work on that, how the transgenic may just been used, and also lignin content is reduced in case of that. So these are some of the references and where actually cell wall hydrolyzing enzyme, which can help for when the cell wall is one of the main concerns, particularly in another. It is hydrolyzed cellulose into a bisaccharides or disaccharide, whatever. So that will be help for the easily for production of. That's why the wood can be very widely containing lignin, cellulose, and hemicellulose. We can convert this 6-carbon cellulose and 5-carbon hemicellulose into sugars. Those sugar can be easily converted. So for this process, there are already people working on that. They have developed some enzyme also, and also they can convert the sugars like a starch with the help of amylase enzyme into sugar. So like that, they're doing a lot of cell wall hydrolyzing. This I think I explained already. The contribution of biotech in a crop sustainability. One is the increase the crop production, how people can be done. And next, help in conserve the electricity because biodiversity and avoid the deforestation. Then reduce agriculture eco footprint, carbon dioxide emission will be reduced. When you go with the biofuels, fossil fuels can be reduced. Now, this is one of the example that because of fewer herbicide, insecticide application of in case of maize in America, they reduce the fuel use. They reduce the carbon dioxide. Equivalent to removing 11.9 million cars from the road. So much of carbon dioxide emission is reduced when you use this biofuels. So that's how it helps to mitigate climate change. And this biotechnology also helps for the poverty and other things. I explained it already. Cotton is the one. I think. Genetic engineering of the microorganisms. Fermentive microorganisms. So genetic engineering also improve that. The one of that is called engineering Saccharomyces cerevisiae. It is a yeast for production of cellulosic ethanol. Now Saccharomyces use only the sugars, then convert sugar to ethanol, but it cannot use the polymers like cellulose. So polymer, then we can modify it such a way that they can also use this. This is a way we can also get cellulose ethanol feeding. The yeast or bacteria is genetically modified, then taken into a bioreactor fermentation. The output of carbon dioxide can be used in soft drinks. Ethanol or butanol used as a fuel, and water can be recycled back, either to grow the crops. So this is a typical picture explained in US. So how the people are producing bioethanol. So using the transgenic plants. This is actually herbicide, pesticide resistance. Transgenic plant gives you more yield, and this yield can be converted into ethanol. This also again the soybean. The varieties of soybean have higher oil amenable. We can also screen the variety which contain high oil content. So those can be done for that. And also this is actually herbicide resistant soybean. They use it in Europe and America. And when you come to the entrepreneurship development, there's a lot of scope. I hope the youth or even the faculty, you can promote them and support the student to make them an entrepreneur. So one of my, around 10 of my students now become a good entrepreneurs, providing job for others, not only in biofuel, they are working in different fields, like molecular biology. The top reason why entrepreneurs should go for biofuel, one, world want to reduce the waste and recycle more. That is one of the reasons why you should go for entrepreneurship in case of biofuel production. Next, there is a growing preference for cleaner and eco-friendly fuels. I told you can just collect waste in urban or rural area and regenerate it as a waste to energy. There's a lot of fund available from the government of India. We can think on that and take those funds. We can make a group. In beginning, it may be difficult, but later stage, you can generate a lot of money also out of the waste to wealth, and also we can provide job opportunity for so many others. The more country want to reduce the dependency on product. This is one of the main India. We are importing more than 80% of the oil, petrol, diesel. If you just reduce it, and not only we can support our country, we will support for the farmers, and also we can, the dollar price is goes down, then only dollar price, otherwise it is very difficult. Today the dollar price is increasing 82.5 or something. It is only because of import of crude oil from other countries. If you stop this, no doubt our price, rupee will be higher than the dollar or equal to dollar. So that was the things earlier before importing the fuel. The price was so nice. Price was rupee was in a better position than the dollar. So today you are going down because of the dollar. The biofuel India is a strategic importance for ensuring India energy balance and also security. It aligns well with the ongoing initiative of the government as a Make India and Swachh Bharat Abhiyan, where you can get a lot of funding for this. Ability to integrate with the ambitious target of doubling of farm incomes. It also helps the farmers to get more money when you have a biofuel. And import reduction, I told you recently. Employment generation, waste to wealth creation. There's a lot of creating win-to-win model for people. The profit, and not only people profit, but also profit to the planet and earth greenhouse gas emission. So thank you so much. Love your environment. Only way to save it, and do plantation every year during your birthday. So I think that will be remembered for you, and also give remuneration also of land time. I started planting from 8th standard onwards. Today I have worth of more than, because not only for benefited me also, it is also my brothers. That's also they're getting benefit out of it. Planted every year. That's more than 5,500 trees, and also some forest trees, around 200 forest trees. Like that is benefited. Recently, I also got some benefit out of the tree I planted, and the teak plant I did it. I got it and cut down and used for our own house. So this is how you can love your environment, and just think on now, because it's giving a lot of benefit to you. You should also think on environment, clean environment. So thank you so much. Now I'll over to the organizer for any question from the audience.
Thank you, sir. Thank you so much, sir, for excellent session, sir.
Thank you. Thank you, madam. Any questions?
Yes, sir. Yes, sir. Yes, sir.
Chat box is. Oh, very good question. Why biofuel not bloomed so much in India compared to other developed countries? Want truth? Sir, the cost of production is already reduced, sir. No problem. I think how to reduce the cost of production, it is already taken care. And now I told you, no, 60 rupees to 70 rupees per liter of biodiesel, but it's not in that. Okay, so that is not the issue. Okay, the first question is, why the people not supported? It's a good question, but still out of the box, not as a thing. I will come back to Vicky. I will answer to this question first. Why the people not doubted? Why I'm telling, the crude oil, even the petrol and diesel price is less in Japan, sorry, in Pakistan, Bangladesh, but it's more in India. The reason is the tax which are involved in that. Number one, the people of particularly the politicians or whatever, maybe maybe bureaucratic, I don't know what is exactly. I will not go much about it. The people not to support that. Why they're not supporting? They'll get a lot of benefit by importing. They have some agreement with the company, oil supplying companies. That's the reason why they are not supporting. Number one. And number two, now the government is supporting compared to previous government or the entrepreneurs. Now government is supporting. Recently, Modi has given a lot of money. The money has given to for boosting biofuel in India. They know that. The truth knows to me very late. I told you very clearly when I was the MSc master student, the nowhere in the picture America. Today America is in number 1 in bioethanol production because they know the value and started working on that. Now the latest stage, now the India is also aware about it. Now the people are thinking on that. Now they've done the blending and others. It takes some more time. Always you will accept little late compared to other countries. Okay, Vicky, I think write the question, please.
Sir, one more participant, sir.
Yeah.
Shall I read, sir?
Yeah, please.
Even though biofuel is economically beneficial, why we cannot use it as a basic fuel for a daily usage when compared with other countries?
No, we can use, for example, methane production. We can do it, no problem. No will constant for that, number 1. Number 2, the ethanol is under the excise duty. Actually, whatever the liquor which are made today, whatever the sarai or whatever the rum, whiskey, everything blend with the this alcohol, ethanol. Ethanol, the people feel that it may be deviated and it is under control. Even for example, I was in sugar factory. In that factory, we were producing ethanol. But even though that they are called as ethanol means 100% pure, it is ethanol. If it's 10% blend with ethanol, then called as rectified spirit. It is called as RS. The RS is used for production of liquors. You are not able to even as a owner of the factory, not even able to take 1 liter of RS for their own purpose. They have to take a permission from the excise. The product is under the excise control. Now there's a lot of liberation now take place. It is moving on how to use that. I think awareness started in around 1996. I started the awareness. 1987 or 90, it is my topic for seminar, ethanol as a fuel. So during my student life, then now it is taking catching area. Is there that? Okay, next question. Abhilash, Dr. Abhilash.
The transition to electronic vehicles and China's business monopoly in even production along with OPEC groups, renewed push to arbitrage fossil fuel, particularly utility has made biofuels politically unavailable alternative.
No, I told you the policy was there. There's a policy. Now government has taken this initiation also. And second one, even though you have that ethanol, because of the tax and otherwise, the cost of ethanol is very high. But still today, for research purpose, laboratory purpose, whatever the other purpose, or a painting industry or nail polish, whatever the cosmetic industry, all require ethanol actually. Painting, cosmetic industry, even cough syrup. But the ethanol which are also produced in US, they are doing in China is very cheaper than us. That is only the technology outbreak. Maybe they have very good mass production technology, less labor, more conversion. The next transition to electrical vehicle, now India is promoting. Probably that will take further. Again, when you come to the electrical vehicle, think of you need energy, electricity. How to get electricity today? Today you are getting electricity only through the 80% through the coal. But when the coal is not exhausted, then you have to again depending on that is there anyway. Electrical vehicle is going on. Probably that takes some time. Internal combustion engines with the BS compatible for 100%. BS6 is compatible. What you said is correct. It is also possible.
So one more participant, sir. Why corn only is the main production for the biofuel?
No, apart from corn also you can do that. But corn content, the grain yield is very high. And you'll get more carbohydrate per unit area. You calculate how much carbohydrate or how much protein the per unit area. 1 hectare of rice. We're getting a lot of yield was so high. That's why the corn is the one. And second one, corn is C3 plant and more energy efficient. It take a lot of sun energy and fix carbon dioxide and get a more food grain for us, corn grain. Then you'll get a more corn grain is, I think we get a more carbohydrate and also protein. I told you already how to produce ethanol out of use. That's why the corn is the main source, and short term also. It is not, it is take around some period, and sugar cane, I think, is 12, 14 months. Whereas corn can produce within 3-4 months. So advantage in the corn, and corn can grow throughout the world, but sugarcane cannot grow. Sugar can grow only in a tropical belt like India, Africa, Brazil, South America, not in North America. That's why USA not promoting sugarcane, because they cannot grow sugarcane in their area. Even Europe cannot do. So some extent only the developed, the tropical belt area only can grow the sugarcane. See the global map, see the tropical area. In that area only sugarcane grow. That's why the people prefer corn, because corn can be shorter, high energy conversion. Solar energy to grain is more energy conversion, and is a C3 plant. Very fastly fix the carbon dioxide. Next, the sugar content is also high. You can use any crop, not only corn. You can. Recently in India, we also started in Ikrisat, Hyderabad. They started producing the sweet sorghum, sweet corn. There is a crop called sweet sorghum. Only the stem can be used, but India, we have abundant sugarcane. No need to depend on the sweet sorghum.
Thank you, sir.
Thank you. Thank you, Sheila, Dr. Sheila Madam, and also thanks to the others for giving
Sir,
up. Thank you so much.
Sir, last one question, sir.
Yeah, please.
In this kind of biofuels, the pollution is low or high?
Pollution is very, very low. That's why I'm telling it is eco-friendly. Please, sometimes what they're telling, the efficiency little less. 10% is there's no problem at all. The carbon dioxide emission, everything is very low. When you blend ethanol, it is an oxygenating agent. Oxygenate meaning high burning. When you burn efficiently, the emission will be less. The carbon dioxide emission will be very less. That is an advantage.
Yes, sir.
The future, what is the rate of biofuel? It is not much, man. Compared to fossil fuel, I'm telling, the petrol goes beyond 500 rupees another 5 years. This biofuel never goes beyond 100, less than 100. Very cheaper. Actually, 1 liter of ethanol is only 40 rupees. You know what is the cost of petrol? Niresh Kumar. What is the rate of petrol? Cost of petrol? You can unmute. Ask a question.
Actually, he's in our hall, sir. He's in our centralized hall.
Yeah, you are correct. 101, 104, depending on the state. But actually ethanol is only 40 rupees per liter of ethanol. Only the problem is excess commodity. That's why you cannot leave it. The people not leave to you. They'll put another prices like that. The actual production cost even less than 40. It is 30 rupees. In Karnataka, there are 2-3 units. Also in Tamil Nadu, also there. Now, Government of India promoting to produce lot of ethanol. They're giving a lot of subsidy also. If you want to start ethanol, they'll give around 5 to 10 crores. The project is 100 crore project usually. Whatever it is there, if you go to 100, out of that you will get around 5 to 10 crores subsidy also.
Yes, sir. Thank you, sir. Thank you so much. On behalf of PG and Research Department of Biochemistry, Sacred Heart College, thanks for you, sir. Thank you. Thank you so much, sir, for wonderful, excellent session, sir. Thank you.
Thank you, Dr. Niyaz. How are you? So when you come to Bangalore?
Thank you, sir.
Nice.
Thank you.
Okay, so nice. I am actually admission officer also. Little bit busy. That's why I just not able to. For the university admission officer now. Apart from university, and also I'm heading university agri entrepreneurship. We got heading agri innovation center. Got around 20 crores funding also now. We are waiting.
So happy to hear from you. Sir, we wish to meet you, sir, in your laboratory. If
Sure,
you
sure,
permit,
sure.
we will
Yeah,
wish
you
to
meet
meet you.
me. Yeah, give me time. Send me your email. Yeah. Yeah.
Sure, sure. Thank you, sir. Thanks, sir. Thanks for coming.
Thank you. Thank you, Father.
Thank you.
Dear participants, dear participants, now the last session will start. Now I invite Dr. K. Prabhu, Assistant Professor, Department of Biochemistry, Sacred Heart College. Hand over the session to you, sir.
Thank you, Sheila, ma'am. Good afternoon, everyone. Once again, I welcome you all to the second day, 8th session of the session, Science Academic Virtual Lecture Workshop on recent advances in microbiology and biotechnology. This is Dr. K. Prabhu from PG and Research Department of Biochemistry, and I will moderate this session. The session is about plant microbiome, looking at the future, and the session is presented by Dr. T.K. Adhyas, Director, South Asian Nitrogen Center, New Delhi. Co-Director, Science, UKRI, GCRF, South Asia Nitrogen Hub, and Professor of School of Biotechnology, Kalinga Institute of Industrial Technology, Bhubaneswar, Odisha. Before we start the session, as a moderator, I have a few instructions to the audience. Kindly mute your audio and video during the session. Ask your question through the chat box. Now I hand over the session to Dr. T.K. Adhyas.
Okay. Yeah.
Sir, one second, sir. I will make you that post. Then only you can able to present. One
yes,
second,
yes,
please.
yes, yes,
One-
yes. Yeah. Can I start?
Yes, sir. Yes, sir.
Okay. So, good afternoon, everybody. I am again back here. I will be talking now about the plant microbiome. In the earlier lecture, I have mentioned about the soil microbiome. And this time we will be talking about plant microbiome with the futuristic outlook. Now, let us begin this with the definition of plant microbiome and all, and what is its significance. So the means like anybody on this earth, any organism on this earth, Plants are also teeming with microorganisms. There are several portions of the plant which have got specific microbial population. They even invade the internal tissues of the plants, apart from those on the external surfaces, on the leaf, on the stem, on the flower, all these places. Now, combined with this vast diversity of microorganisms in the soil rhizosphere, that's a separate region, of course, the root region. And we coined a specific term of rhizosphere. We will come to that. Rhizosphere has been coined, I think, almost more than 100 years back, 1904. Hiltner, he introduced the term rhizosphere to specifically indicate the microorganism associated with the root region. Now, this plant soil associated microbes comprise the plant microbiome. This is actually involved in the plant health and other serves as a reservoir of additional gene. This is the most interesting part, which I'll be talking to the people, to the colleagues, that plants can access when needed. The thing is that there are a lot of organisms involved in this thing. In fact, there have been a lot of work on the microbial association with the plant, but as a plant microbiome research together, putting as an exclusive terminology, plant microbiome, or the plant associated bacteria or microorganisms, not only bacteria, of course, fungi and other fungi is also there, archaebacteria is also there, virus is also there. So this thing came with the sudden spark in knowledge of the human microbiome. You must have heard or aware of this. What is that human microbiome? The microorganisms associated with the human being, both the inside and the surface of the human being. And that has been found out that these
Adhiya, sir? Adhiya, sir? Star is inside the link only, but Star is inside only, but unable to talk. What, ma'am? Ma'am.
இல்ல நான் மெசேஜ் அனுப்பிச்சிட்டேன் சார். சார் அதியா சார்?
Yes, sir. I think Sir is having some technical issues.
Yes, sir. Star is having some network. Star is inside the link only, but unable to talk. Adhiya, sir?
Sir, kindly you make a call, sir.
Okay.
No, I think it was muted. Can you hear me now?
Yes, sir. Yes, sir.
I'm getting you, sir. So
Yeah,
just- okay.
I changed the connectivity my end also, thinking that it is giving trouble.
Okay. No, sir. You can proceed, sir. Please, sir.
Okay, I don't know. Is it visible? No, I have taken upload, I think. I have taken share. Okay, all right. No, I think this got disconnected.
Sir, you need to present again, sir.
Yeah, I have to share again, I think.
Okay, sir. Proceed. And now it's working, sir. Started screen sharing.
It's working
No,
all
that's fine.
right? Okay,
Okay,
now
okay,
let
sir.
me see whether it moves or not. Yeah,
Okay,
right.
sir.
Okay, good. I changed my connectivity also this end to another one so that there is no problem. Yeah. Now
Okay,
the now I was talking about the plant microbiome. The microorganisms and the plant relationship, or the microbes from the plant atmosphere context, has been worked for quite long time. But still, as a microbiome assembly for the plant, the interest has come up only in recent times, only after vigorous work on human microbiome. You know that almost 1/3 of our body weight, human being body weight, is of the microorganisms, and they play a great role in our daily life, even keeping us healthy, happy, and whatnot. That the microorganisms are concerned. And with that idea in mind, these people started concentrating on plant microbiome also, to find out the role of microorganisms on the plant health, and also the other impact on the plant. Now, understanding the regulation of plant trait expression, hence the plant performance, and how this is impact ecosystem function, requires that we study the impact of the plant microbiome. Now, thinking about the soil microbe plant interface, I think, which is a basic requirement to find out the impact of microorganisms on the isolated from the soil and applied to the plant, is not new. I have mentioned this earlier. However, the concept of the plant microbiome perceives this interaction more along the lines of microbe soil, microbe plant, microbe interface. A little bit of confusing attitude application you might be thinking, rather than making it straightforward like soil microbe plant interface. So now, what is the difference? I'm talking like that microbe from soil, microbe from plant, and the interface between the two. So the difference is understanding that the microbe is comprised of genomes vastly more complex than that of the plant alone. It is very interesting that they have a genome which is different from that of the plant. And the plant genome and the microbial genome, they have got a lot of interaction. The nature of this interaction, actually, these genomes serve as an extension of the plant's genetic compendium. That means it may not be, when one sequence the plant genome, they may not have any microbial genome in that plant genome, but this is extended as a genetic compendium of the plant, is associated with the plant, effectively coined as the plant's second genome. That means the additional genome for the plant in action. Now, this picture, it is a model of the plant microbiome with emphasis on the below ground habitat. Now, you can see here, there are two parts of this picture again. On the right hand side, you have got plus. On the left hand side, here is a minus. Now, plus is that where you have got mutualistic interaction, positive to plant growth and this thing, what is that called, health, like plant growth promoting traits, protection from the pathogen, and the systemic resistance, both induced and acquired. I will come to that later, definitely. Now, There is on the negative side is the pathogenic interaction, where the microorganisms have caused disease onto to the plant, increased disease response, decreased biomass, the plant dries up, nutrient content is reduced, and then finally die or whatever it is. That means the negative impact on the plant growth. Now, this plant actually, one thing is that they release carbonaceous substrates. That is what is popularly known as the plant exudate. Now, these plant exudate, they help, they support thriving of microorganism on the surface of the plant, what we call a root region exclusively. That is what is called as the rhizoplane. And there is also the bulk soil microbes. Some of these substrate, they leak into the bulk soil, will come subsequently. And that bulk soil microbiome is also there. And there is inside the plant also, there are some microbiome, which is known as the endophytic microbiome. So basically, these are the different types of microbiome partitioning into. You have got also some of the microbes, they release some exudates, some compounds they release, which I will tell you. And there's a complex interaction in that case. So, focusing on the below ground habitats within the plant microbiome, the subsections of bulk soil, which is the major part of the soil, there is a microorganism usually in the range of 10 to the power 6 to 10 to the power 9. Rhizosphere, it may go even up to 10 to the power 10 or so. Rhizoplane, this is a very specific group of organisms which remain on the exclusively attached to the surface of the root region, not the rhizosphere, but on the surface. Rhizo stands for root, on the root surface. Epiphyte. Epiphyte is that on the above ground, this thing, plants. It could be on the stem, as I said. It could be on the leaf. It could be on the leaf. We all know that term, phyllosphere, microorganism. On the seed, that is the spermosphere. Now, this is on the above the plant surface, and the other one is that inside the plant, that is the endophytes. Okay. Now, this is the diagram. I think a very old diagram of the plant and the microbe plant habitat for the microbial life. You can see here there is the rhizosphere, the root region, the rhizosphere, this one. And then inside the plant is the endosphere. These are the bacteria on the leaf surface. Okay. Now, before we start talking about the microorganisms they perform, first we must know how to study them. Okay, so this is actually the bacterial, the different microbial communities, and they can be isolated, they can be studied both through cultivation method, whereby you grow them on a culture medium, on petri dishes, and then put it into different media and check for their functional potential. But only thing hitch is that, as I told in my earlier lecture, only minority is culturable. Now, the other half is that without cultivation. Without cultivation means here we don't use any culture media or anything like that. Rather, we this thing extract the DNA from the source, from the sample, maybe rhizosphere sample or the plant sample or whatever is that. Then undergo different omics processes like metagenomics, metatranscriptomics, metaproteomics. This I have told earlier in the lecture, in my earlier lecture. And then subsequently we go for identification of the community analysis and phylogenetic, using phylogenetic and functional markers, and then group them. What are the group of organisms? So this is the basic. Now this almost all the microbial analysis, whether it is from the plant, animal, human being, everywhere, this has become almost a norm that we follow this omics principle, by which we can know more as compared to that of the cultural or the cultivation practices. Now, the functional importance of the plant microbiome. What are the functional importance? Some of these are already, quite well known. That is the mobilization or acquisition of the nutrients, nitrogen, phosphorus, different micronutrients, zinc, etc. They are mobilized and acquiesced by the plant with the help of microorganisms. This plant microbiome associated with the rhizosphere, they help in the mobilization and acquisition of nutrients. The second importance is that of the out-competition and antagonism of pathogen. Now, this is a very interesting one. There are several microorganisms having either HCN production potential or the allelopathic potential, or even they have antibiosis potential, whereby they inhibit the growth of the pathogenic microorganism, which would have otherwise affected the normal growth of the plant. Plant stress alleviation, they support the plant to grow under stress condition, specifically, say, for example, drought, then that moisture stress, excess of moisture, flooding, or heavy metal stress, and etc. Even these are the different plants are subjected to stress because plants have to be rooted to the soil, and they cannot, if the temperature increases, they cannot go get into the AC room. So these microorganisms, they help to alleviate this stress. Plant growth promotion, actually, this is by the production of different hormones and all that they help the plants to grow, like indolastic acid or the gibberellic acid or GA. They produce, and with the help of these hormones, there is a promotion in the growth of the plant. And the next is the co-metabolism. They perform several co-metabolic functions, and degradation of toxins, some pathogen toxins and other things are also produced by the this microorganism. Now, this together actually has become to be known as the plant growth promoting bacteria, PGPB. I mentioned PGPB because there is also PGPR, that means which are the rhizospheric microorganism. But this is a broad group, plant growth promoting bacteria. Of course, now the question comes that there are also several other things like mycorrhiza, which are not bacteria. They are also helps in the growth promotion. So this group of organisms, they help in the promoting in the growth. And then it, of course, this is a quite well-known, almost nearly 200 years that we have the knowledge of this group of organisms, although in a different form, but we were knowing that they were having some beneficial effect on the plant. Now, in this figure, we are just trying to show the conferred by the rhizobacteria and their products. You can see here on the left-hand side, the topmost this thing portion, plant growth promoting rhizobacteria. There's a model plant, actually. This is Arabidopsis thaliana, just a drawing of it. They produce some molecules and releases it, this PGPR, which actually results into the growth of the plant. And then that is the basic principles of plant growth promoting rhizobacteria. That is the positive impact of the PGPR, where they produces growth hormones, they mobilize the nutrients, as I have mentioned earlier. Then the also several other things they help, and then those are the direct and positive impact. The other one is that you can see that it has covered the plant in a bubble. It is protecting from other things like pathogenic microorganisms are coming and being bounced back from this bubble, outside the bubble. This is the PGPR here. These microorganisms, they release some molecules which help the plant to grow and avoid these pathogenic interactions. Many of them are the plants are thereby are protected. Now, when we take these together, both the nutrition and growth as well as the resistance and tolerance, we can see that some of the things have got dual role, like say for example DAPG or bioiodine or the different nod factors. These have got growth promoting impact as well as the means pathogen inhibition mechanism, like salicylic acid or the different types of compounds are listed here. And this side we have got the indolactic acid, which is popularly known to result into the growth of the root and the shoot, and also opaline effectors. These are the different type of compounds which have helped directly in the, but in the center, I mean the group of compounds, they are like bacterial volatile organic carbon, acetoin, or the nod factors. These nod factors actually help in the recognition of the rhizobium onto the root region. And these are the different things which are help in the growth. They confer benefits to the plant. And now I will just a little bit of endophytes I will mention. This is the rice endophytes. This has been found that in the inside the root region of the rice, there are the endophytes. And this is actually, they can help in the contribution of the nitrogen fixation and plant growth promotion. There are specific characteristics of the endophytes. And they involve actually, this figure is a bit complicated figure with so many things are mentioned here. They colonize the inside the cell, endophytes, inside the tissue, mostly in the cortical region they stay. Also sometimes they're found in the sarcovascular tissue also, flagella, pili, etc. Through that they attach to the plant tissue. And then inside the plant, they take up some sugars, what are the plant synthesizes, or degrade some of the aromatic compounds, even oxidize the methane, and then ability to cope with the oxidative stress. And in this process, they help the plant with certain production of certain things like nitrogen fixation, siderophore formation, or even the ACC deaminase enzyme production, which actually hydrolyzes ethylene. And there is the it results in the inhibition of the ethylene, not hydrolyze, sorry. It results into the inhibition of the ethylene production so that the plants can grow. As and you know that ethylene has got a negative impact on plant growth and development. So these are actually, in addition, also there is also the oxidative stress avoidance and detoxification also they help. And then plant growth promotion like ACC deaminase. These are all mentioned. I mean, both ways, different type of terminologies are used, but basically they help in these endophytes. Many of these endophytes, they depend on the sugars produced by the plant, or sometimes the other aromatic compounds they degrade, and they help the plant with the nitrogen cycling, especially N fixation, then nitrification and denitrification, etc., etc. And they help the plant to grow. Okay, these are the endophytes. Now I will give another example of the rice plant under high CO2, elevated CO2. Now here you can see this is a normal ambient CO2, normal plant biomass and yield. And when you grow it in a under high CO2 concentration, then what happens is the plant grows. It is actually called as the CO2 fertilization effect, that carbon dioxide helps into the more of photosynthesis and the plant becomes taller, although very little increase in the yield is there. But also there is at the same time high methane and nitrous oxide emission is there. Now this this below ground, you can see the root also. There is a lot of root, higher activity, and then this result into the more release of the root exudate into the soil region, and then there is the high activity of methanogens, etc., etc. But they are characterized by the high root biomass. Now, whereas the physicochemical properties of the soil types determine the composition of the soil biomes. I mean, in the soil, bulk soil. The biome, soil biome, when I talk, I mention about the microorganism in the bulk soil. Soil biome as well as a plant root exudate. These are determined by the physicochemical properties of the soil types. The plant root exudates can create an environment at the rhizosphere that gradually alters the soil biome for the selection and establishment of a rhizobiome. That means only a specific group of organisms are selected because of the continuous providing the photosynthete in the form of root exudate. Not only photosynthete, some of the amino compound and small compound, simple compounds are also provided by the through the root exudate. These exudates, together with the root immune system, would finally select for those microbes that, due to further adoption, have evolved mechanism to colonize the root rhizoplane, or even sometimes inside the root tissue, that is the endosphere, which are the endophytes, they finally grow. So the major driving factor is the root exudate. So now, I mean, we can talk about that, the currency of the microbiome. I mean, like we have all our currency in our pocket, we have got rupees, and with rupee we can go anywhere in India. So this is the currency of the microbiome. The microbiome, they take the they are means grow with at the expense of the exudates released by the plant. But that is not always a negative impact on the plant, but they also support the plant. That's what I mentioned, is that exudates in the soil rhizosphere, they are chemicals released from the plants or from microbes, mostly organic acids and then amino compounds, etc., etc. Through exudates, plants are able to communicate with the microorganisms. Sometimes some small flavonoid compounds are also released through the root exudates, and they help the plant, the bacteria, to check the to contact, I mean, communication with the microorganism. I can give you a example of a flavonoid compound which is released through the root exudates, and the rhizobium. They have it activates a nod factor into the rhizobium, and the rhizobium attaches onto the root region, and thereby it starts, after that it enters into the plant root and develops the nodule. Of course, that's the second part, but the first part is that it calls from the bulk soil, wherever rhizobium is there, it sends messages through the root exudates, please come here. Here I am standing, waiting for you, and you can come and put your grow in association with me and develop the root nodule. So this is about a specific example, but otherwise, the elicit assistance in environmental acclimation to alleviate stresses like pathogen attack, then a drought limiting nutrient acquisition and metal toxicity. These microorganisms benefit from plant exudates by using them as a resource, in many cases carbon, but also other nutrients, as I was mentioning, that are small amino compounds, as well as the different flavonoid compounds. We have analyzed many of these root exudates in rice plants, and then found that especially acetic acid is one of the important compound released in the root exudate. Through the exudate currency, the plant microbiome serves as an extension of the plant genome. Now, these microorganisms, the gene that are present in these microbes associated with the plant or the plant microbiome, they serve as an extension of the plant genome. So some sort of genomic interaction is there between the plant and the microorganism in the rhizosphere. So if you talk about the root exudate and the microbial interaction, plant uptake and release, they help the root exudate and microbial interaction. They help in the nutrient mobilization and their uptake by the plant. Microbial uptake and release, microorganisms take up some of the compound from the root exudate, and they produce a certain specific compounds and release it for the plant to interact. Impact on the plant function, this root exudate, they have got impact on the plant function, because certain amount of root exudate has to be released into the surrounding areas of the root. Sometimes it can go almost 40% of the photosynthate that the plant fixes carbon. 40% of that photosynthate is released into the root region as root exudate, depending upon the scenario, and the impact on the bacterial function. So both ways, this root exudate, they help in the release of, because of the sometimes acidic pH, they will result into the release of phosphorus and other things, and then the plant can take up. The microorganisms can grow. A microorganism take up this root exudate, and thereby they fix the nitrogen and other mobilize the other nutrients. So this influences both the plant function as well as the microbial function. Now, importance of the microbiome to plant genomics and phenomics. I mean, how this microbiome, they influence the plant genomics and phenomics. Now, the plant microbiome have been implicated in the mediation of over a dozen of plant functional traits, like the root, I mean, the size of the root, then vegetative, then regenerative, stem and leaf traits, how the stem will grow or the leaf will grow, their pattern. So it follows the extremes in plant trait expression observed in the majority of invasive plants, including traits of physiology, then leaf area allocation, shoot allocation, growth rate, fitness, and biomass. They likely involve help from the microbiome. However, this type of impact actually across broad phylogenetic and biogeographic ranges needs to be explored. I mean, these are only some cases, only it is known, very few cases rather, especially some of the crop plants like rice and then some of the legumes, these are known, but not for everybody, because on an average, I think we have got around 50,000 species of plants on the earth, and there is a lot of work to do on this. Now, you can see here, I think we come back to a similar type of figure where the interaction determining the plant phenotype and trait. You have got the environmental factors, bright sunshine, then lot of rain, and this influences the environmental factor. The agricultural management practices, you sometimes, I mean, the farmers sometimes spray the insecticide and pesticide and all the different agricultural management factor. Now, all this influences the plant to release the root exudate in the root region. And you can see these are the root region. There are the microorganisms which are present. Now, sometimes the farmers also apply the microorganisms as inoculant, microbial application. And these are majority of these features are affected by the plant genotype and plant domestication. Now, this plant genotype is actually influences the plant, identifies the plant type, and the type of the plant it grows and all, and the domestication results into the identification of a specific variety type or varietal growth, which much probably better than that of the wild type and all. And then all these things result into the expression of the plant phenotype. Now, this is actually the complex interaction between the plant. Now, if we see this here, interaction between the plant, fungi, and bacteria in the rhizosphere, you can see here, we have got the, here we try to introduce fungi also in the black circle, not circular ellipse. Then they actually, this is the bacteria, they releases the different signaling molecules. These signaling molecules actually, they influence the signaling molecules like auxins and cytokinins. They result in influence the plant growth. Okay. Now, these plant growth, because of the growth, now the root exudates will produce the root exudates, chemicals and branching factors, they will come and influence, and there will be interaction between them. Now, the root exudates also contains phytochemicals and the organic acid, and it picks up the microorganism. It's a model of a bacteria with a flagella, some sort of thing, like something like that. They actually produces lot of signaling molecules like peptidase and DHL2, AL1, this type of things. And this interaction, they result into the quorum sensing also also synthesizes the quorum sensing molecules, and this actually helps in the and these bacteria again, the signaling molecules for the secondary metabolites they produce, and they help into the establishment of the plant. And then that's how the entire interaction between plants, fungi, and bacteria in the rhizosphere is determined. So if we look back, the functions of the plant microbiome are essential for the host. I have not gone into the detail of PGPR and other type of things, which is now have become staple. All the friends, young friends and students, they all know about this. They have learned about it. So if we look back, the functions of the plant microbiome are essential for the host. We have seen that how they help. Specific enrichment of microorganisms in plant-associated communities exist. We have seen that there is a quorum sensing is there, and they result into the selection of the specific group of microorganism in the root region. I had another figure, I don't know where I have kept it, where with a different color code, the microorganisms are shown, and there is this, I mean, to specifically mention that from the bulk soil, specific group of microorganisms are selected by enrichment, and they get associated with the plant. The origin of plant-associated bacteria is different. It varies. I mean, it is not the same that from another plant. It could come from another plant. It could come from the bulk soil. It could come from the other practices. So, contaminant factor also, that the plant-associated bacteria, they are for each and horizontal gene transfer contributes to plasticity and evolution of plant-associated bacteria. There is a transfer of the genes, like the plant has got certain genetic elements, and the bacteria has got plasmid and all. There's a possibility of the transfer of these genes from the bacteria to the plant, which can then get integrated and result into the positive features for the plant. Now, horizontal gene transfer, they contributes to the plasticity and evolution of plant-associated bacteria. Now, we need availability of various nutrients. I'm trying to repeat some of the things so that it can become more clear. Going to the availability of various nutrients and surfaces, the plant-soil interface is also considered a hotspot for horizontal gene transfer process via plasmids. I was mentioning you the plants, the bacteria may have plasmid, and in the plasmid some genes can be there which can be transferred onto the plant. The recent progress in microscopy tools, so you have got now several types of microscopy like atom force and all, where we can find it actually track the bacteria and then find out where it is moving and how it gets attached onto the plant root, and then enters inside the plant cell, gets settled into the endosphere of the plant. So these things are actually our knowledge is developed, and then we come to know about all these things. So the basic thing is that what I want to mention is that horizontal gene transfer contributes to plasticity and evolution of the plant-associated bacteria. Now, what is the future of this plant microbiome research? I mean, how they can help. Now, some of the things are already, as I mentioned earlier, that is the role of microorganism in the sustainable agriculture through the development of the microbial inoculant. We have used several microbial inoculants as biofertilizers, like nitrogen-fixing bacteria, P-solubilizing bacteria, or the zinc-solubilizing bacteria, K-solubilizing bacteria. These group of organisms are used as biofertilizer, which go into the plant, near to the plant or the rhizosphere, and then mobilize the nutrients and supply the plant with those so that they can grow normally. Biocontrol is that they produces certain metabolites, as I have mentioned, like either like HCN or the antibiotic compound or allopathic compound, whereby they force the pathogenic organisms away from the plant. So this way they can they help the plant to thrive difficult scenarios of pest. I mean, in a microbial attack, which can cause disease-causing organism, or even stress protection products. The different compounds they may, like ACC deaminase, I have mentioned. ACC deaminase, it helps into the production of the different oxidizing ROS productions, and then that can be controlled, and then especially under drought condition or under where there is a stress, moisture stress, they can result into the formation of reactive oxygen species, ROS. And these microorganisms, they produce the different compounds like another type of oxidized. I mean, they can control the oxidation potential and maintain the plant tissue in a much better condition. So these stress protection products, specifically for all the biotic, all the abiotic stresses. Two biotic stresses, of course, also are there. Abiotic stresses and then heavy metals, etc., etc. Now, in the future, the plant microbiome will have a greater importance for plant breeding and plant biotechnology. How? Until now, the primary plant pathogens were considered in three approaches. In these approaches, however, it is suggested that beneficial aspect of the entire microbiome should be integrated as a biomarker. That means take the entire microbiome, what are the total beneficial aspect, or a package of beneficial aspects should be integrated as a biomarker. Now here comes the question, the concept of the synthetic microbiome. Synthetic microbiome is that it is actually they get two or three major important positive impacting bacteria, and then result into the and inoculate the plant. This group of this artificially synthesized consortium of microorganisms, they can go and get settled and then provide a multifarious beneficial impact on the plant. Now, the plant microbiome can be an important source shaping source shaping other microbiomes. As I said, that they can influence the other microbiomes to go away, and only the specific ones are selected. Like, say, for example, the Pseudomonas and the Bacillus species, Bacillus and Penibacillus also, they are very exclusively selected in the rhizosphere region of some of the crop plants. So this type of specific selection is there. This meaningful overlap of phylogenetic diversity can be recognized among microbiomes which are in some way linked to each other. And this may also include the human habitat and plants. Sometimes the residues and other things which are incorporated into the soil. From there also these microorganisms can go. Like, say, for example, different pathogenic bacteria, they can land into the through the soil. They can land in, I mean, through the drain, I mean, sewage and all. When the sewage sludge and other things are dumped into the soil, they can get accumulated and in the root region, and they can sometimes be selected for the plant in the plant rhizosphere. So this is a, like, I can tell my own example. While we are isolating, trying to isolate some of the P solubilizing bacteria, we found a mycobacterium, which is a mycobacterium, you know, that what is that called, tuberculosis is causing organism, but not that all mycobacterium are tubercular. But we found a mycobacterium species which has a very high P solubilizing activity. Now we try to find out, because you cannot use a organism which has got potential for contamination. So we checked it, and then we found that it might have come from certain human related contamination into the root region of the plant. So this is also possible that they can come from other ways. Now let us take this figure here. The plant biome, the plant microbiota environment interactions together. And then you have got the start with the below ground interaction, that is the rhizosphere. You can see here different color coded dots are there representing the different microorganisms. And these actually this plant microbe interaction at the strain level, there is a mechanistic understanding. That means how they will invade, how they will attach onto the root region and all. And then some of them may go inside and then remain in the as the endophyte. On the surface of the surface also, you have got the organisms, phylosphere. Some of them may be dangerous also. These are the pathogenic, different red color, this thing, pathogenic microorganism causing disease to the plant. Now here, this is the interesting is that some of these microbes, they influence the, they produce a certain volatiles and result into the carrying the messages to the crop plant. Now, these are the management of the plant microbiota can be through biofertilizers and biopesticides. I was telling you, and also at the same time, there is the plant microbiota in stress adaptation or response. I was talking you about the plant disease control. I think there, as you might be knowing, that there are two types of control. One is the systemic acquired resistance in the plant, and one is the ASR, adapted systemic resistance. Now, these actually, most of these are mediated by certain volatile compounds, like ASR is the salicylic acid mediated thing is there. And the plants, what they do is, these microorganisms, what they do is that they synthesize salicylic acid that is carried through the plant, volatilized, and then this is synthesized when there will be attack from the pathogen. This is then volatilized and surrounds it areas it goes. Like that in a colony, some houses thief has come and you go south, thief, then everybody become very aware. So here also the plants also through that type of mechanism, through the connection with the combination with the plant microbe, they synthesize the compound salicylic acid and then it is volatilized to the plant region. The other plants become aware, yes, the pathogen is coming, so let us be ready. So this type of acquired systemic resistance also, these plant root related microorganisms are involved. Now, selection of plants for efficient interaction of with the plant microbiota. Now, the one thing is that plant breeding program have gone through selection. They select only specific and improved plant type. The plant breeders are not that much bothered about the microbiota and all, but now I think probably they are trying to know about this in more detail. Many cases, this results into the loss of plant genes. If you see the wild type of rice and the cultivated rice, the total genomic length is almost 1/3 less as compared to the wild type of this thing. This has been proved for other crop plants also, while wild type plant or wild relatives have got a much more total genome content as compared to the cultivated types, because the plant breeders, they go on selecting the plants for better features, especially in terms of the yield and other quality parameters. But in this process, many of these things are lost. This has also happened when many of the important crops were domesticated. I mean, you know, earlier over the years, the nature has resulted into the growth of the plants, and then we have picked up, oh, this plant has got this property, so we picked up and started growing and establishing this. This is what is called the domestication. Now, these not only resulted in loss of genetic plant diversity, but also, to example, if you take a normal growth promoting parameters, and then we have to add it. We have recently conducted an experiment wherein we have isolated the wild type of rice, wild and weedy rice, from some swamp areas and all, and also some scented rice variety, local scented rice variety, them with the modern yielding rice cultivars, and then found that the modern yielding rice cultivars has got a very few plant growth promoting bacteria, because they know we know that we will apply the fertilizer, we'll have pesticides and everything to ensure the growth and yield of the crop. But for the wild type varieties, they grow wild and normal this thing and under different stress. So the micro associated in the root region or inside the endophytes, they all have got their setup to withstand that. When we pick these wild type varieties and then plant selecting the best type, all these things are lost. Now, plant breeders should consider the associated microbiome within the holobiont, the plant itself, to confer additional plant traits to modify them. Now, future perspectives and information gaps. Although the plant microbiome is recognized as an intense treasure trove of microbial diversity, numerous important crop species and their natural relatives have not yet been studied for their associated bacterial communities. I mean, we do not know each and every one of them. A few of them have definitely been studied, but not that we know each and every plant. With an approximate number of 500,000 plant species, a lot of work lays ahead of the plant microbiome research to explore new aspects about phylogenetic diversity of plant-associated microorganisms in the future. I mean, this may be specifically interesting with the future adverse impact of climate and the environment that we are going to face in near future. Already we are facing, and it may be more intense, and probably the answer lies with those microorganisms, especially with that of the old types and all, which have ruled or established on this earth for several million years, those weedy or the wild type of plant, and then find out what is the reason, what is the plant microbiome impact for them to thrive for such a long period, and then utilize them for protecting our resources from the adverse environmental impact. I think this is the last slide. Yeah, thank you very much. So this is what I wanted to mention. About the plant microbiome, which is now caught the fancy of the people. Initially, when we started plants separately, isolated the microorganisms separately, started to put them as an inoculant. Then find out afterwards, we went further to find out what the inoculant, how does it help the plant. It fixes the nitrogen, then how the nitrogen is transferred. So we went back, but the nature has made the microorganisms in such a way that they help the plant, they have a natural relationship with the plant and allow the plants to thrive. So we have to go back to that and then find out what is the exact mechanism that the plants are, that this bacteria or the microbiome is doing to help the plant to grow better. Thank you very much.
Yes, sir. Thank you, sir. Thank you for your excellent presentation about the plant microbiome. I hope participants are highly benefited. Sir, we have a lot of questions, sir, in the chat box.
Very
Can I
good.
read, sir?
Sure.
So the question is, what is the role of pesticides on plant biota?
What is the role of?
What is the role of pesticides on the plant biota?
Yeah. See, this we apply the. I would like to make the question more generalized instead of sticking to pesticide alone. Any agricultural practices that we undertake while growing a crop influences the microbial relationship. That could be even plowing the field. That could be adding the organic manure. That could be adding the green manure. that could be flooding the soil, that could be applying the pesticide. Any of the agricultural practices, and a lot of studies has been done how they are the microbiome, especially the rhizosphere and the bulk soil microbiome are influenced. So this is a known fact that they influence the microbial diversity by the different agricultural practices. And they actually vary from the group of organism, because some of these organisms which are capable of breaking down the aromatic compounds, say for example for pesticide, for which the question was asked, I mean those may be selected under those scenarios to grow. Actually, we find that the actinomycetes, actinobacteria, now we don't call them as anymore actinomycetes. Actinobacteria, they predominate, they are very hardy organisms. They predominate in those situations and then help to decontaminate the toxic residues.
Thank you, sir. The next question is, which soil layer has the most microbes?
This is, yeah. You want to continue some of part of this question?
Can microbes be swapped between plant types and still have the same function?
That's what I think I mentioned in my lecture, is that it is not clearly known exactly, but we find that there is a difference among the microorganism. What bacteria or fungi or this thing, it will be selected into the root region. It depends upon, there is a intricate relationship between the plant and the microorganism to be selected. Like you give, I have a house, I want to give somebody on rent. So I find out whether that fellow is dependable, whether he will give the rent in time and all. This type of inquiry we make. So the plant also makes that type of inquiry. Bacteria also makes that type of inquiry, that he will not ask me to leave after 6 months. So that type of, or he will give me the not stop the plant water supply. So here water supply means the root exudate. The root exudate should continue. So the plant and the bacterial association is very specific, and it cannot be, probably it cannot be generalized that okay, all bacteria, these bacteria will be there throughout. But we have found some keystone microorganisms which are present in several rhizosphere regions. I mentioned about Pseudomonas and Bacillus species, also Penibacillus. These are quite common in the rhizosphere.
Thank you, sir. Next question, how to adapt to changing the climate by targeting the microbial community?
Yeah, that's a very nice question. There has been microorganisms have been found to thrive under adverse climatic conditions, and these these type of microbes have been selected. Like I can give you example of the microorganism which can help the plant to grow in a drought situation where the less of moisture is there. The bacteria, they synthesize a polymer, polybeta hydroxybutyrate, or PHB. This PHB, it forms a film onto the clay mineral, onto the clay, entrapping some water. So that means, in other words, it helps in the retention of water for a certain period of time, allowing the plant to withstand the drought to a certain extent. So this is one of the example I can give. And the plants, microorganisms, they are in their, they have got lot of potential in the different climatic scenarios. They can help and allow the plant to grow.
Sir, one request, sir, from morning session, soil microbiome, we have some questions, sir. Shall I ask?
Please.
From first PG, Asina asked, is there any possibilities to regenerate soil with the help of microorganism in a radioactivity affected area?
Oh, very interesting question. I think you must have read about Chernobyl, this thing. Have you?
Yes.
Who has asked me, I don't know whether she is there or not. But
Yes,
so Chernobyl was, you know, that there was a melting down of the reactor, and then there was a leakage of radioactive element, and that area was completely restricted. After this thing, almost 30 years or so, now they find that plants started growing little bit. So this is, there are some microorganisms you might have heard about its name, Deinococcus radiodurans. This Deinococcus is a very interesting bacteria. It can withstand almost 300 times of radioactivity as compared to the human being. And say that means it can withstand the atomic explosion. After that also it can withstand. This is a very peculiar bacteria. One of the major impact of the radioactivity is that our DNA gets broken into pieces. I mean, I'm trying to be as simple as possible. So in Radiococcus radiodurans or Deinococcus radiodurans, this DNA as usual, that also gets broken down because of the high radiation. Now, after some time, they join themselves and again the genome is rejoined. The bacteria thrives well. So this is this type of organisms, they are there, and then they can help the soil to recover. I mean, not that it's billions of bacteria, once they grow, they will die. They will add the organic carbon into the soil, and then thereby help the soil to rejuvenate. So it is possible. I mean, what I want to give this example of Deinococcus radiodurans is that microorganisms are there which can influence and then help the soil to rejuvenate.
thank you, sir. So one more question from second MSc Bhuvaneshwaran asked. So please explain what are the four-dimensional objects of soil?
Yeah, four-dimensional object, as you said, normal 3D, you see, if you take a soil block, there's a length, breadth, and height, and the width. No, the 3D, normal 3D concept. Now, along with that, and then here we have got the soil, water, soil bulk soil, that is the materialistic, and the soil gas or the air in the soil. Now, along with that, we also have a fourth dimension, that is the interaction aspect. The interaction for all the different things, they together, they form the fourth dimension of the soil, and that actually influences how the soil functions. I mean, any soil, say for example, a garden soil or a desert soil or a soil from a very, different type of areas. Now, the difference will be between these soils will be their fourth dimensional aspect, or the interaction between the different soil components, soil microorganisms, both the biotic and abiotic factors together, they result into the expression of the soil property. So this is what is the fourth dimension.
Thank you, sir. I think no more question.
Okay.
Sir, am I audible?
Yes.
Okay, sir. Thank you, sir. Thank you so much for your excellent presentation, sir. Also, special thank to Dr. Adya, sir, for
Thank you.
accepting changes in schedule change.
Yeah. Okay, it happens.
Thank you.
Thank you very much. So I am leaving. Thank you.
Sir, right, sir, please, sir, please.
Yes. Yes, Dr.
Thank you
Niels.
for wonderful sessions, sir. Excellent.
So thank you very much.
Thank you, sir. Sir, and yes, now I invite Dr. K. Prabhu, sir, and Niyaz Ahmed, sir, the section.
So, dear participants, we have come to the end of the 2-day Science Academic Virtual Lecture Workshop. Participants are requested to fill the feedback form, and the e-certificate will be posted to your email. On behalf of the PG and Research Department of Biochemistry, Sacred College, Tirupattur, we thank all the eminent speaker, convener of the workshop, Dr. DJ Bakiraj, Coordinator, Dr. I. Niyaz Ahmed, Head of the Department, faculty members, and beloved participants for making this workshop a grand success. Once again, a special thank to Dr. D.K. Adya sir for accepting changes in the schedule. We shall meet you again for another kind of academic program soon. Thank you and God bless you all.
Thank you, Adya sir. Thanks for your availability.
Thank you, sir. Thank you, sir.
Thank you, sir.
Thank you very much. I'm quitting.
Thank you, sir. Thanks, sir. Thank you, sir. Thanks for your availability.